Hello and good day everyone. So it's me Chekgu online, and basically today we're going to learn about the lock and key as well as the induced fit mechanism for the enzyme reaction. So before we begin with the discussion of this topic, I hope that you can subscribe, like, and also share this video. Okay everyone, so before we begin to learn about the mechanism, we all have first to be familiarized with the structure of enzyme and also the substrate. So enzyme is a biologically active substance that catalyzes chemical reaction by lowering the activation energy barriers and converting the substrate molecules into the product, while the substrate is a reactant in a chemical reaction. As we can see in this diagram, so for the enzyme, it has the active site. So active site has a unique geometric shape that is complementary to the shape of the substrate. As we can see here, the shape is complement to each other, meaning that the enzyme specifically react with only one or very few similar substrates. For instance, if the enzyme is sucrase and then it will react with sucrose to produce the glucose and also fructose. Right, so we are going to learn about the first model, which is the lock and key mechanism or lock and key model or lock and key hypothesis. This is a very popular model. So it consists of 4 steps, but first we have to know which one is lock, which one is the key. So the substrate is the key while the enzyme is the lock. So the first step is that the substrate that has a complement shape to the active site of the enzyme will actually bind to the enzyme, bind to its active site, forming the enzyme substrate complex. And then once they have bind and forming the complex, they will actually produce the product. So we call this third step as enzyme product complex. So once the product has been produced, they will actually leave the active site of the enzyme, and the enzyme will be made available to bind with another substrate. So we make example. For example, we have sucrose and sucrase. Sucrose is the substrate and sucrase is the enzyme. So sucrose will bind to the sucrase, forming the sucrose-sucrase complex, and then the product for this what we call enzyme and substrate is the glucose and fructose. So once the glucose and fructose has been produced, the glucose and fructose will actually leave the active site of the enzyme, and the active site of the enzyme are made available to bind with another sucrose, as this enzyme is sucrase. Alright, so this is the second mechanism, which is the induced fit mechanism or induced fit model. So the mechanism is just the same as the lock and key mechanism. However, there is a little improved, what we call improvisation on this model. So as you can see here, we also have the substrate and also the enzyme. The same thing, substrate is the key while the enzyme is the lock. So substrate will actually bind to the active site on the first step. However, as you can see here, on the second step where the difference happens, the substrate will actually determine the final shape of the enzyme, meaning that the enzyme is slightly flexible. So enzyme will actually change the shape slightly as the substrate bind to produce the enzyme substrate complex. So once they have bind, so the product will be produced to produce the enzyme products complex. So once the product has been produced, the product will actually leave the active site of the enzyme, making the enzyme available to bind with another substrate. So they are actually the same thing with the first model, which is the lock and key model, but it differs on the second step, as which the final step or what we call as the substrate itself will actually determine the final shape of the enzyme. And that is the difference between the induced fit and also lock and key hypothesis. That is the end of the topic. So we have discussed what is enzyme, what is substrate, and how actually enzyme binds to the substrate. And also, we have learned about the two mechanisms, which are the lock and key mechanism or lock and key hypothesis, and also the induced fit mechanism. So I hope that you can spot the difference. The difference is just on the second step, of which for the induced fit model, the substrate will determine the final shape of the enzyme. So that's it for today's topic. Thank you for listening. And please study smart, don't study hard. And do not forget to subscribe, like, and share this video and also this YouTube channel. So thank you very much, and we'll see on the next video. Assalamualaikum.